Welcome to the deep dive. Now, if you've ever, cracked open a biology textbook, the story of how a cell dies usually seems pretty straightforward, almost binary.
Right, you've got your two main categories.
Exactly. On one side, there's classic necrosis, that's the accidental death, messy, uncontrolled, maybe after trauma, and it kicks off a whole lot of inflammation.
The collateral damage scenario.
And then on the other side, you have apoptosis. That's the cell's clean, programmed self-destruction, almost like surgical removal. Quiet, orderly.
The neat and tidy exit.
But the deeper scientists dig, the more they realize that nature, well, it's rarely that simple, is it?
Definitely not. It loves complexity.
Our sources today are really pulling back the curtain on something pretty revolutionary. This idea that processes that look accidental, that look like necrosis, can actually be highly regulated, genetically controlled events. We're moving way beyond that simple binary.
Into a much more nuanced landscape.
So our mission today is to unpack this specialized demolition crew. We're doing a deep dive into these programmed non-apoptotic pathways. They're critical for survival, for defense, but also, unfortunately, they pop up in almost every major disease. We're talking necroptosis, pyroptosis, ferroptosis, and that fascinating double-edged sword, autophagy.
Yeah, that one's particularly interesting.
And why this matters to you listening is, well, it's pretty simple. Understanding these molecular switches, these different ways a cell can choose to die, that's fundamental. It's the key to developing the next wave of targeted therapies for everything from cancer to stroke, even neurodegenerative disorders.
That really is the critical shift in thinking we need to grasp. For decades, if a cell swelled up, burst open, spilled its contents, and caused inflammation.
It was just necrosis,
No.
passive, accidental.
Exactly. Just, bad luck for the cell, an unavoidable consequence of injury. But what our sources really hammer home is this concept of regulated cell death. Mechanisms that look, for all the world, like that accidental mess.
But they're not accidental at all.
Not at all. They're controlled by specific genes, specific signaling pathways. It's programmed demolition, even if the result looks chaotic.
That changes the whole game, doesn't it?
Completely. Because now we're looking at a whole suite of pathways, all genetically hardwired, that give the cell choices, agency in how it responds to specific kinds of stress. And just to underline how fast this field is moving, the pathway we call ferroptosis, it wasn't even formally recognized, didn't have a name, until 2012.
Wow, that's really recent in biological terms.
It is. So we're right in the middle of this massive expansion of knowledge, realizing the cell has way more levers it can pull to decide its fate, even when facing really severe injury.
Okay, let's start unpacking this group. Maybe we begin with the one that has perhaps the most confusing name, necroptosis.
Necroptosis, right.
The name itself tells you something's different.
It really sounds like a hybrid. It looks like necrosis. The cell swells, organelles rupture, the membrane breaks, inflammation follows.
All the hallmarks of messy, accidental death. But mechanistically, underneath the hood, it operates with the precision of a programmed pathway, more like apoptosis.
It's got a defined signaling cascade.
So the key distinction we need to keep in mind here is that necroptosis is often called caspase-independent programmed cell death. Apoptosis, the clean pathway, relies heavily on those specific executioner enzymes, the caspases.
Right, caspases are the classic executioners in apoptosis.
Necroptosis seems to kick in specifically when those caspases are blocked or somehow out of action.
That caspase independence is absolutely central. It helps explain its biological role. So let's trace the signal. The best studied trigger, the one we know most about, starts outside the cell. It involves the ligation, the binding of the TNFR1 receptor. That's a tumor necrosis factor receptor 1.
Okay, TNFR1, I know that one, it's usually involved in inflammation and it often triggers apoptosis too, right?
Exactly. Normally, it might push the cell towards apoptosis, but under the right conditions and critically, if those apoptotic caspases are inhibited for some reason, the cell gets diverted, it takes the necroptosis off-ramp instead.
So the same starting signal, but a different path depending on whether the main road, apoptosis, is blocked.
Precisely. Once TNFR1 is activated, it acts like a docking station. It recruits this crucial multi-protein platform inside the cell. And this platform involves two key kinases. Think of them as molecular switches. They are receptor-interacting protein kinase 1 and 3, RIPK1 and RIPK3.
RIPK1 and RIPK3. Okay, kinase added phosphate groups to things, right? They switch other proteins on or off.
You got it. They sense the signal from TNFR1, get activated themselves, and then pass the message along by phosphorylating downstream targets.
Okay, so we have the external trigger, TNFR1. We get the RIP kinases recruited and activated. How does that internal molecular switch flipping translate into the cell literally blowing itself apart? That seems like a big jump.
It is, but there's a key player that bridges that gap. Once RIPK3 is activated, its main job is to phosphorylate another protein hanging out in the cytoplasm. This one's called MLKL. That's mixed lineage kinase domain-like protein.
MLKL.
This phosphorylation of MLKL, that is the final irreversible command in this pathway. Once MLKL gets that phosphate tag, it dramatically changes shape. It activates and then starts clumping together with other activated MLKL molecules. It forms these large, structured oligomers.
So it goes from being a single protein to part of a big complex.
A big, dangerous complex. Because these MLKL oligomers then physically move, they translocate from the cytoplasm right up to the inner surface of the plasma membrane.
Ah, okay. Getting close to the edge.
And they don't just sit there, they actually insert themselves into the membrane. They basically form these unregulated pores or channels. Imagine punching thousands of tiny holes all over the cell's outer boundary.
The structural integrity is gone.
Completely compromised. Water rushes in uncontrollably down its osmotic gradient, the cell swells up like a balloon, and boom, it ruptures. So you see, it's a highly regulated, specific molecular cascade, TNFR1 to RIPK13 to MLKL phosphorylation to membrane insertion that results in the messy, inflammatory death we associate with necrosis.
Wow, okay, that sounds like a biological nuclear option. It's messy, it causes inflammation. Why would evolution bother developing a programmed pathway like this when it already has the clean, quiet apoptosis option available? What's the advantage?
That's a great question. It functions primarily as a critical backup system, particularly against pathogens, especially viruses.
Okay, how so?
Think about certain clever viruses, like cytomegalovirus, for instance. They've co-evolved with us for millennia, and they've become masters of manipulating host cells. Many have developed specific proteins inhibitors whose sole job is to shut down the host's apoptosis pathway, usually by directly blocking those key executional caspieces.
Ah, so the virus tries to prevent the infected cell from committing suicide quietly.
Exactly. If the virus succeeds in blocking apoptosis, the infected cell can't self-destruct cleanly. It becomes a replication factory for the virus.
So necroptosis is the cell's plan B.
Precisely. It's the host's countermove. If the caspases are blocked, the cell says, okay, fine, we'll use a different pathway. It activates the RIPK-MLKL system instead. By triggering necroptosis, the host ensures the infected cell still dies, limiting viral spread. Yes, it's messy, but...
The mess itself is useful. The inflammation.
Absolutely. That inflammation is a vital distress signal. It calls in the cavalry, the immune cells, to deal with the infection site. So it kills the infected cell and raises the alarm.
That's fascinating, a sort of scorched earth defense. So it's a contingency plan against viral tricks. But what happens when this system gets activated inappropriately in diseases that aren't infections? What are the consequences then?
Well, when necroptosis goes haywire, the consequences can be severe precisely because you get that combination of cell loss plus inflammation. We see this pathway playing a major role in ischemia-reperfusion injury.
That's the damage when blood flow returns after a blockage, right? Like in a stroke or heart attack.
Exactly. When the blood rushes back in, carrying oxygen, it paradoxically causes a burst of oxidative stress and inflammatory signals. This can trigger necroptosis in cells that might have otherwise survived the initial lack of oxygen. It kills potentially salvageable tissue.
So it contributes to the damage after the main event.
Yes. And beyond acute events, necroptosis is strongly implicated in chronic conditions too. Things like acute pancreatitis, severe liver diseases like steatohepatitis, fatty liver disease with inflammation, and importantly, various neurodegenerative disorders. Parkinson's disease is one where it's heavily studied.
Which implies a potential therapeutic angle.
Huge potential. If we could develop drugs that specifically inhibit RIPK1 or RIPK3, we might be able to prevent a significant amount of cell death and inflammation in these conditions. It's a major focus of drug development. And just to be complete, it's not always pathological. Our sources mention it plays a normal physiological role too.
Oh, really?
It actually occurs naturally during the formation of the mammalian bone growth plate in development. Shows it has constructive roles too, likely in tissue remodeling.
That discussion of inflammation and backup systems sets the stage perfectly for our next pathway. Pyroptosis. If necroptosis is the caspase-independent rupture that acts as a backup, pyroptosis seems to have a slightly different mission. It's less about bypassing a blockage and more about deliberately shouting for help.
That's a great way to put it. The name itself gives it away, pyro, referencing fire or fever.
This really is the explicitly inflammatory programmed cell death pathway. Unlike apoptosis, which aims to be immunologically silent, pyroptosis is defined by the massive, rapid release of inflammatory mediators, the most famous being that fever-inducing cytokine interleukin-1 or IL-1.
Right. This pathway is firmly in the territory of the innate immune system. It's typically switched on when microbial products, bits of bacteria or viruses, or sometimes danger signals from damaged host cells get detected inside the cytoplasm of a cell, often an immune cell like a macrophage that's been infected.
So the cell knows it's infected or something is seriously wrong internally.
Yes, and the cell needs to do two things urgently: die itself to stop the pathogen from replicating, and sound the alarm as loudly as possible.
Okay, how does it achieve that double whammy?
The key initiating machinery is a complex protein scaffold called the inflammasome. Think of it like a modular alarm system that isn't pre-assembled. It snaps together rapidly inside the cell only when specific sensors, these cytoplasmic innate immune receptors, detect a threat.
Like detecting bacterial flagellin or viral DNA.
Exactly. Those kinds of pathogen-associated molecular patterns, or sometimes host-derived danger signals. Once detected, these sensor proteins trigger the assembly of the multi-protein inflammasome complex.
So the cell senses the invader, quickly builds this alarm platform, and what does the assembled inflammasome do?
Its primary job is to act as an activation platform for a specific executional enzyme, caspase-1.
Ah, another caspase. But you said this isn't apoptosis.
Right. This is where it can get confusing because the name caspase is involved. But caspase-1's role here is very different from the apoptotic caspases like caspase-3 or necrosis-7. Caspase-1, which also used to be called interleukin-1 converting enzyme.
Okay, that name is suggestive.
Very. It performs a crucial dual function. First, it finds the inactive precursor forms of IL-1 and another related cytokine, IL-18, which are just sitting in the cytoplasm, and it cleaves them. It cuts them into their biologically active mature forms,
Unleashing the inflammatory signals.
instantly. These active cytokines, potent pro-inflammatory molecules and pyrogens, fever inducers, are then rapidly released outside the cell. That's the shouting for help part.
And a second function, what does caspase-1 do to the cell itself?
At the same time, caspase-1, along with some related inflammatory caspase like caspase-4 and MyNH5 in humans, targets other proteins. Specifically, they activate pore-forming proteins, particularly one called gasdermin D.
Gasdermin D.
Activated gasdermin D fragments then go to the plasma membrane and form very large pores, much like MLKL in necroptosis, but through a different mechanism involving caspase-1. These pores disrupt the osmotic balance, water floods in, and the cell undergoes endolytic death. It swells and bursts.
So similar outcome to necroptosis swelling and bursting, but triggered by caspase-1 and the inflammasome, and tightly linked to releasing IL-1.
Exactly. The cell bursts open, releasing not only the active IL-1 and IL-18 it just processed, but also all its other intracellular contents, which are themselves often inflammatory. Things called DAMPs, damage-associated molecular patterns. It maximizes the local inflammatory response.
It's a really interesting evolutionary choice. Why go for this explosive, seemingly energy-intensive, highly inflammatory death? Apoptosis would also kill the infected cell, wouldn't it? What's the strategic advantage of pyroptosis?
Speed and mobilization. Apoptosis, being quiet and contained, is relatively slow. Often, the apoptotic cell gets neatly cleared away by neighboring phagocytes before it spills its contents, minimizing inflammation. That's great for normal tissue turnover or removing single damaged cells without fuss.
But not ideal for a raging infection.
Not at all. In an acute infection, the priority isn't quiet tidiness, it's rapid containment and recruiting massive reinforcements. Pyroptosis achieves this perfectly. It quickly eliminates the pathogen's replication niche and simultaneously floods the zone with the strongest possible chemoattractant signals, like IL-1, to bring in neutrophils, macrophages, and other immune cells very, very quickly. It sacrifices local tissue tranquility for rapid systemic immune defense.
Okay, let's shift gears now to ferroptosis. You mentioned this is the newcomer on the block, only really defined around 2012.
Relatively speaking, yes, a very active area of research.
And if necroptosis and pyroptosis focus on protein kinase cascades and caspases leading to membrane rupture, ferroptosis sounds much more chemical.
It is fundamentally a chemical process, yes.
You defined it as an iron-dependent pathway triggered by excessive lipid peroxidation. That sounds like the cell's fats are essentially going rancid, driven by iron.
That's actually a pretty good analogy of fats going rancid. The name Ferroptosis highlights that dependency on iron, specifically ferrous iron, Et2 plus RO. We need iron for lots of biological processes, but free iron is also a potent catalyst for generating reactive oxygen species, ROS, free radicals.
The damaging byproducts of metabolism.
Exactly. And the core mechanism of ferroptosis boils down to the cell losing its fight against oxidative stress, specifically stress targeting its lipids. Every cell has antioxidant defense systems. A major one relies on a molecule called glutathione and an enzyme called GPX4, which specifically neutralizes lipid peroxides.
Okay, so glutathione is the protective shield.
It's a key part of the shield. Ferroptosis happens when the oxidative damage, often catalyzed by high levels of intracellular iron-generating ROS, simply overwhelms that glutathione-dependent defense system, particularly the GPX4 enzyme.
So the shield fails, the iron acts as an accelerant, and the oxidation runs rampant. What exactly is getting oxidized?
It's the fats specifically, the polyunsaturated fatty acids, or PUFAs. These are crucial components of all the cell's membranes. The outer plasma membrane, mitochondrial membranes, ER membranes, you name it.
They make the membranes fluid and functional.
Right. But they're also vulnerable to attack by ROS. In ferroptosis, you get this runaway chain reaction of lipid peroxidation. The ROS, kicked into high gear by iron, attack these PUFAs, breaking them down into toxic byproducts and destroying the structure of the membrane.
And if the membranes are destroyed?
If the cell is destroyed, membrane integrity is essential for everything maintaining gradients, transport, signaling. Widespread lipid peroxidation leads to a catastrophic loss of membrane function, increased permeability, and ultimately cell death. But crucially, even though it looks like destructive chemistry,
It's still regulated.
yes, it can be specifically prevented. If you use iron chelators to remove the excess iron, or compounds that boost the glutathione GPX4 system, or vitamin E type antioxidants that directly stop lipid peroxidation, you can block ferroptosis. That proves it's a distinct, regulated pathway, not just random oxidative damage.
You also mentioned earlier that ferroptosis leaves a specific fingerprint, something you can see under an electron microscope that distinguishes it.
Yes, its ultrastructure is quite unique, particularly concerning the mitochondria. While necroptosis and pyroptosis involve plasma membrane rupture, ferroptosis hits the mitochondria in a characteristic way.
How so?
The most striking features are the loss of the mitochondrial cristae. Those inner folds disappear or shrink dramatically, and often a rupture of the outer mitochondrial membrane. The mitochondria look shrunken and damaged in a very specific pattern. This distinct mitochondrial morphology is a key diagnostic feature for identifying ferroptosis in research.
Given its reliance on iron and oxidative stress, where do we see ferroptosis popping up in terms of disease?
It's implicated in many conditions where those factors are known to be major players. Neurodegenerative diseases, for one, Parkinson's, Alzheimer's, Huntington's. There's often evidence of iron dysregulation and oxidative stress in affected brain regions.
Okay.
Also, ischemic injuries like stroke, similar to necroptosis, the reperfusion phase creates that burst of ROS that can trigger ferroptosis. And maybe most excitingly, in cancer.
How does it relate to cancer?
While many cancer cells have altered metabolism, often high iron requirements, and sometimes specific vulnerabilities in their antioxidant pathways, this potentially makes them more susceptible to ferroptosis than normal cells. So there's huge interest in developing drugs that induce ferroptosis specifically in cancer cells.
Turning the pathway against the cancer.
Exactly. It represents a completely different way to kill cancer cells, especially those that have become resistant to traditional apoptosis-inducing chemotherapy. Controlling this iron-lipid axis is definitely one of the hottest areas in therapeutic development right now.
All right, let's move to our final mechanism. And this one, autophagy, seems to present the biggest conceptual puzzle. The name itself, from the Greek for self-eating, tells you it's about the cell consuming its own parts.
Cellular recycling.
But we often hear about autophagy in contexts like fasting, longevity, cellular cleaning. It sounds like a good thing, a survival mechanism.
And primarily it is. That's the crucial point to start with. Autophagy is first and foremost an evolutionarily conserved survival mechanism. It's a highly sophisticated adaptive response to stress.
So it only becomes linked to cell death under extreme circumstances.
Generally, yes. It's that dual nature survival strategy versus potential death pathway that makes it so complex and, frankly, fascinating to study.
So let's talk about the survival role first.
Yeah.
When does the cell ramp up autophagy?
The classic trigger is nutrient deprivation. If a cell is starved of amino acids or growth factors, autophagy kicks into high gear. It allows the cell to essentially cannibalize non-essential components, breaking down proteins and organelles to generate building blocks and energy, just to keep basic functions going.
Tiding itself over during lean times.
Exactly. It recycles internal resources. Beyond starvation, it's critical for cellular quality control. It acts like the cell's garbage disposal system, clearing out damaged proteins, protein aggregates, which tend to build up during aging and in diseases like Alzheimer's or Parkinson's, and also getting rid of old or damaged organelles like worn-out mitochondria through a selective process called mitophagy.
Keeps the cellular machinery clean and functional.
Right. It prevents the accumulation of toxic junk.
Okay, so it's a complex, multi-step recycling process. Can you walk us through the basic steps? How does the cell actually eat parts of itself?
Sure. It starts with initiation. Cellular stress, like low nutrients, activates specific signaling complexes that kick things off. This leads to step 2, isolation membrane formation. A unique double-layered membrane structure, kind of like a flattened sac, starts to form in the cytoplasm. This is called the phagophore, or isolation membrane.
Where does this membrane come from?
The exact origin is still debated, but a major source is thought to be the endoplasmic reticulum, the ER.
Okay, so this double membrane starts forming. Then what?
Step 3 is elongation and cargo capture. This phagophore membrane expands and curves around, engulfing a portion of the cytoplasm. This can be bulk engulfment, just grabbing whatever's nearby, or it can be selective, targeting specific things like protein aggregates or damaged mitochondria that have been tagged for destruction. This whole process requires the coordinated action of a whole family of proteins encoded by autophagy-related genes, or ATPase genes.
Dozens of them.
More than a dozen key ones, yes. And as this membrane bag elongates and closes, a specific protein called LC3 gets modified. It gets lipidated, linked to a lipid called PE, and incorporated into the membrane.
And LC3 is important because
Lipidated LC3 is essential for the membrane expansion and closure, and it remains stuck to the completed vesicle. This makes it the gold standard marker that researchers look for to see if autophagy is actively occurring in a cell. If you see punctate dots of LC3 under a microscope, you know autophagosms are forming.
So LC3 is the tag that says recycling in progress. What's the final step?
The final step is maturation and degradation. The fully enclosed double membrane vesicle, now called an autophagosome, travels through the cytoplasm and fuses with a lysosome.
The cell's digestive organelle, full of acids and enzymes.
Exactly. When they fuse, they form an autophagosome. Inside this combined vesicle, the lysosomal enzymes degrade the captured cargo, the proteins, lipids, organelles, breaking them down into their basic building blocks, like amino acids and fatty acids. These building blocks are then transported back out into the cytoplasm for the cell to reuse.
A remarkably efficient internal recycling system. Okay, so we've firmly established it as a pro-survival pathway. Where does the death dilemma come in? How does this process, designed for survival, get implicated in cell death?
That really is the million-dollar question, and it's still debated. We know that if the cellular stress is simply too severe or prolonged, autophagy might not be enough to save the cell, and the cell dies despite activating autophagy. That's clear.
The rescue attempt failed.
Right. But there are situations, often involving extreme stress or perhaps genetic manipulation in labs, where cells undergo massive levels of autophagy. They become filled with autophagic vacuoles right before they die. This has led some to classify this as autophagic cell death, implying the process itself becomes lethal.
So the question is, did the excessive self-eating kill the cell? Or did the underlying stress kill the cell, and the massive autophagy was just a symptom of a dying cell trying desperately to survive?
Exactly. Causation versus correlation. Most researchers currently lean towards the latter, that autophagy itself rarely delivers the lethal blow, but rather cell death occurs when the stress overwhelms the cell's adaptive capacity, including its autophagic response. However, the dysregulation of autophagy, whether too much or too little, is undeniably linked to pathology.
Can you give some specific examples of how messed up autophagy contributes to major human diseases?
Absolutely. In many neurodegenerative disorders, the problem seems to be insufficient or impaired autophagy. In Alzheimer's, for instance, there's evidence that autophagosomes form but struggle to fuse properly with lysosomes. The trash bags get filled but can't make it to the incinerator, leading to the buildup of toxic proteins like amyloid beta and tau.
And Huntington's.
In Huntington's disease, the mutant Huntington protein itself seems to directly interfere with the efficiency of the autophagy machinery, again leading to toxic accumulation. So in these cases, boosting autophagy might be beneficial.
Okay, so insufficient autophagy is bad there. What about cancer?
Cancer is where it gets really complex and paradoxical. In some contexts, autophagy can act as a tumor suppressor, maybe by clearing damaged components that could lead to mutations. But in established tumors, particularly in the harsh, nutrient-poor environment deep inside a tumor mass, cancer cells often hijack autophagy.
They use it to survive.
Yes, they ramp up autophagy to recycle materials and generate energy, allowing them to withstand starvation, hypoxia, and even chemotherapy. So in many advanced cancers, blocking autophagy is being explored as a way to make cancer cells more vulnerable.
Fascinatingly context-dependent. What about infections?
Crucial for infectious diseases, it's a direct defense mechanism called xenophagy, eating foreign material. The cell recognizes intracellular bacteria or viruses and targets them for destruction via autophagy. Pathogens like Mycobacterium tuberculosis, which causes TB, and Shigella are known targets.
And we know it's important because
Because if you knock out key autophagy genes, like a G5, in animal models, they become highly susceptible to these infections. Mice lacking at G5, for example, are much worse at controlling TB infection.
And finally, you mentioned a link to inflammatory bowel diseases.
Yes, a very strong genetic link has emerged for IBD, like Crohn's disease and ulcerative colitis. Large-scale human genome studies found that common variations, or polymorphisms, in a specific autophagy gene, ATG16L1, significantly increase a person's risk of developing Crohn's disease. This suggests that subtle defects in how autophagy functions in gut cells contribute to the chronic inflammation seen in IBD.
Okay, we've taken a deep dive into these 4 distinct pathways: necroptosis, pyroptosis, ferroptosis, and autophagy. It's clear that the cell's decision-making process for death is way more intricate than we used to think. Now let's try to synthesize this. The big takeaway seems to be that these pathways aren't operating in isolation, right? They're interconnected, they influence each other.
Absolutely. That interconnection is where a lot of the cutting-edge research is happening now. They're not siloed processes. Think back to that TNFR1 receptor we discussed for necroptosis.
Right, the starting point.
Depending on the cellular context, are caspases active? Is the inflammasome assembled? What other stress signals are present? That single receptor ligation event could potentially lead to classic apoptosis or inflammatory pyroptosis or caspase-independent necroptosis. The cell is integrating multiple signals to choose the most appropriate response.
So it's like a complex internal negotiation based on the specific threat and the cell's current status.
Exactly. And understanding what tips the balance, what makes a severely injured cell undergo regulated necroptosis versus just falling apart through passive accidental necrosis, for instance, that's a major question. Because where there's regulation, where there are specific molecular players like RIPK kinases or caspase-1 or GPX4,
There's a potential target for therapy.
precisely, we gain the potential to intervene, to either deliberately trigger a specific death pathway or, perhaps more often, to block one that's causing disease.
Let's talk more about that therapeutic potential. Because ultimately that's the goal, right? Translating this knowledge into treatments. If we can target these specific molecular switches, what are the most promising strategies emerging?
Let's take them one by one. For necroptosis, remember, it's often a backup pathway causing inflammation. So the main therapeutic goal is usually inhibition.
Stopping the inflammatory rupture.
Right. In conditions like stroke, heart attack, reperfusion injury, or maybe even neurodegenerative diseases where cells are dying via this RIPK-MLKL pathway, the strategy is to block it. Develop specific small molecule drugs that inhibit RIPK1 or RIPK3.
And are those in development?
Oh, yes. Several RIPK1 inhibitors in particular are in clinical trials for various inflammatory and neurodegenerative conditions. The hope is to save tissue that would otherwise be lost during these acute events or chronic inflammatory processes.
Okay, that's about preventing death.
Right.
What about ferroptosis, that iron and lipid peroxidation pathway? How specific can we be there?
The targeting for ferroptosis is potentially very specific, which is exciting. Because it hinges on iron chemistry and the glutathione and GPX4 antioxidant system, you have clear chemical vulnerabilities to exploit.
So 2 angles again, protection and induction.
Exactly. On the protective side, for conditions like stroke or neurodegeneration, you could develop better iron chelators to soak up that catalytic iron, or compounds that specifically boost GPX4 activity or provide vitamin E-like lipid antioxidants, protect the vulnerable cells from oxidative destruction.
And the induction side, killing cancer cells.
That's arguably generated even more excitement, because many cancer cells, especially those resistant to apoptosis, seem to have a heightened sensitivity to ferroptosis. They might have higher iron needs or weaknesses in their antioxidant systems. So researchers are actively developing drugs designed to induce ferroptosis.
Oh, they do that.
by either inhibiting GPX4 directly or blocking the production of glutathione, essentially removing the cell's defense against lipid peroxidation and pushing it over the edge. It's a completely different way to attack cancer compared to traditional chemo.
A chemical attack targeting a specific vulnerability. Now, autophagy, with its dual role, that sounds trickier to target therapeutically. How do researchers decide whether to boost it or block it?
It is definitely trickier and highly context-dependent. You have to know what role autophagy is playing in that specific disease or cell type.
Okay.
So in neurodegenerative diseases like Alzheimer's or Parkinson's, where the problem is often the accumulation of toxic protein aggregates due to impaired autophagy, the goal is generally to enhance the pathway. Find drugs that boost autophagic flux, helping the cell clear out the garbage more efficiently.
Trying to restore the cleaning function.
Right. But in many advanced cancers, where the tumor cells are actively using autophagy to survive stress and resist treatment, the goal is the opposite: block autophagy. Inhibit key Ad-G proteins to basically starve the cancer cells or make them more sensitive to other therapies.
So you need to know if it's helping or hurting in that specific situation.
Absolutely. And the discovery of those specific genetic links, like ATG16L1 and IDD, is really important here. It suggests we might eventually move beyond just generally boosting or blocking the whole pathway towards more precise interventions, targeting specific components or regulation points within the autophagy machinery tailored to the disease genetics.
It really underscores how critical it is to understand exactly which switch controls which type of cellular demise in which context. Whether you're trying to save a neuron after stroke by blocking RIPK, or kill a resistant tumor by inducing ferroptosis, or fine-tune the recycling system in someone with Crohn's.
Precisely. The cell has this incredibly sophisticated toolkit for self-destruction, evolved for host defense, development, and damage control. The modern medical challenge is learning how to safely and effectively manipulate that toolkit for therapeutic benefit.
So wrapping up this deep dive, I think the fundamental message is clear. The old, simple picture of cell death, just accidental necrosis versus clean apoptosis, it's really no longer sufficient. It's functionally obsolete.
The reality is far more nuanced, far more regulated.
The cell's demise is controlled by a range of sophisticated genetically programmed options. Let's just quickly recap the essence of these 4 pathways we explored.
Okay, necroptosis. Think programmed inflammatory rupture. Key takeaway. It's a vital backup against viruses, but also a target for stopping unwanted cell death in things like stroke and neurodegeneration by inhibiting RIPK kinases.
Right. Pyroptosis, this specialized inflammatory death driven by the inflammasome caspase-1 releasing IL-1 implication. Understanding how the body mounts a rapid feverish immune response and finding ways to modulate that when it becomes excessive.
Then ferroptosis. The iron-dependent death caused by runaway lipid destruction. Clinical relevance. Its unique chemical basis offers very specific ways to either protect cells, like neurons, or, maybe more excitingly, induce death in hard-to-kill cancer cells.
Yeah. And finally, autophagy, the complex self-eating process, the double-edged sword. Key implication. It's essential for cellular cleaning and survival, but can be hijacked by cancer. Modulating it, boosting or blocking specific Ad-G genes or steps is crucial for tackling everything from neurodegeneration to IBD to cancer. Ultimately, the cell's fate, whether it bursts dramatically, rusts from the inside out, quietly fades away, or desperately tries to recycle itself back to health, is governed by these distinct molecular pathways. And our ability to understand and, crucially, to control these switches is really what defines the cutting edge of modern medicine.
And maybe that brings us to a final, slightly provocative thought to leave our listeners with, especially thinking about autophagy. We've established it's primarily the cell's own survival mechanism, right? It ramps up during stress to try and cope.
Right, self-preservation.
So the question becomes, when we develop therapies designed to boost autophagy, say in a neurodegenerative disease to clear out toxic proteins, are we always pushing that stressed cell towards survival? Or is it possible that if the cell is already too damaged, if its energy reserves are too low, could boosting its self-eating capacity actually push it over the edge? Could we inadvertently accelerate its demise by forcing it to consume itself even faster when it lacks the resources to benefit from the recycling?
Wow. So trying to help might actually push it towards self-destruction in some cases.
It highlights the incredibly delicate balance, doesn't it? The knife edge between life-saving adaptation and lethal self-consumption that's orchestrated within every single cell. A balance we're only just beginning to truly appreciate, let alone master therapeutically.